Hello everyone, how are you doing? I hope so everyone is well and happy in their life. So, I want to give you a very interesting question to think about. Everybody will give me their point of view on this question. So, the question is why your eyes look so similar to your parents? Have anyone got this compliment from your relatives or maybe from your friends or somebody who come at your home and visit you for the first time? I'm pretty sure some of you have got this compliment that your hairs match with your father, you look so similar to your mother, your lips are exactly like your mother, or you are as polite as your mother, right? We all got such kind of compliment from any random person that visit us for the first time. So why so? I'm pretty sure all of us look very different from our parents. Then why we have such similarities still associated with our parents? Can anyone answer this question? If you're wondering why there is so much similarity, I'll tell you a story. I'll tell you a story today why you have such a similarity. So let's go and learn about a very interesting topic for today, which is known as DNA replication. So, what do you understand by replication? What can be replication? Have you used this word before in your life? Anyone here? So, yeah, replication in general sense means replicating, creating a photocopy of something, right? But what can be this DNA replication? I hope so everybody can see my screen. So, what is DNA replication? As somebody told me, replication means creating a photocopy, right? So, does it mean we create a photocopy of DNA as well? Is it correct or not? You guys will answer me by end of the class. Okay. So, how does a DNA look like? It is twisted. It is helical in shape, right? Every DNA is twisted or helical in shape. It has two strands. Can you see? This is the first strand and this is the second strand, right? These two strands have anti-parallel polarity. What is this anti-parallel polarity? If one strand runs from 3' to 5', another strand runs from the opposite. That is 5' to 3'. That's what anti-parallel polarity means. So these two strands run in anti-parallel polarity. Now, you must be noticing I have drawn the lines in between this helical. These are what? These are nucleotides. How many nucleotides does a human DNA have? Do anyone know? Yes, we have 4 nucleotides. What are the name of the 4 nucleotides? Those are adenine, thymine, guanine, and cytosine. In simpler way, we write it as A, T, G, and C. Right? Now, this adenine is attached to thymine using a double hydrogen bond. This is double hydrogen bonding. Similarly, the guanine and cytosine are attached to each other using 3 hydrogen bonding. So what do you feel? Which bonding is stronger? Adenine thymine or guanine cytosine? Yes, definitely the bonding between guanine and cytosine is very strong comparing to adenine and thymine. So, if I want to break this DNA, which is helical in shape, if I want to unwind this DNA, I want to take 2 strands and open it up. So, which bond I will target? Adenine thymine or guanine cytosine? Yes, you guys are very smart. Definitely, we will target the bond which is less stronger or which will require less energy. So we will break adenine and thymine bond. But who does that for us? We cannot do that, right? So who does that? For that, we have some enzymes. which we will learn one by one. Okay. So here we go. So the enzyme which is responsible for breaking this DNA nucleotide, which is adenine and thymine, is known as helicase. Now, this enzyme recognizes this place where adenine and thymine are associated to each other by two hydrogen bond, and when it does, it finds actually the origin of replication. So actually it initiate breaking of DNA. We will understand what is this replication slowly, but first learn how exactly it is happening. So this helicase recognizes ORI, which is origin of a replication. The moment it recognizes origin of replication, it creates a replication fork like this. So basically, if I have a twisted something and I break it, it open up, right? So this is known as a replication fork. Just like the fork we use to, eat our food, that kind of fork. So the helical enzyme is responsible to identify the place through which it should start replication, and then it create replication. Is the story end here? No. After this, once we get this replication fork, now the actual story begins. Slowly and slowly, this DNA starts unwinding. So it start opening up. Now, if it is opening up, that means this strand, which is from 5 prime to 3 prime or 3 prime to 5 prime, they now have 1 parental strand. This is parental strand. Now, over this parental strand, somebody is needed who can create a new strand like this. Now, who is responsible for that? For that, we need the DNA replicase enzyme. For that, we need DNA polymerase enzyme. Now, this DNA polymerase have some condition. He says that I will work on only one single strand. And secondly, he says that I need somebody who tell me from where to start. So we said, fine, we have two separated single strand. So you can begin from either 3 prime to 5 prime or 5 prime to 3 prime. But he says, no, I need that parental strand which goes from 3 to 5 so that I can begin from 5 to 3. They said, okay, fine, you can do that. And second problem of the DNA polymerase is he need a help. For that, we provide him another enzyme known as RNA polymerase. Now, this RNA polymerase is the one who start this formation of new strand. Now, once RNA polymerase is attached from 5 prime to 3 prime polarity, now they keep on adding adenine, thymine, guanine, cytosine, and so on. It keeps on adding the nucleotides. Suppose parent DNA have A, that is adenine, so it attach a thymine. If it has a guanine, it attach a cytosine. Like that, it keeps on happening without any break. That's why this particular newly formed DNA is known as leading strand. Just because it is not breaking in between, it is continuously going, it is continuously leading. So what do you think? This stop here? No. The whole DNA open up and the leading strand keep on going. So here you notice that one strand is parental and another is a newly formed strand, right? Now similarly for the other part of the DNA strand, The problem is here DNA polymerase starts from behind. Because do you remember the condition? It said that it needs a parental strand which is from 3 prime to 5 prime so that it can start from 5 to 3, right? So what happens? It goes like this, 3 prime to 5 prime, and then again 3 prime to 5 prime. But when it looked behind, it noticed that a lot of DNA is opening up, but it is not able to continuously form the strand. So who come in to help? RNA polymerase again. So RNA polymerase says, I am there. I will initiate the strand making and you keep on attaching the nucleotides. The only problem is that they are in fragments. These fragments are known as Okazaki fragments. Now this Okazaki fragments keeps on attaching to the nucleotides. Say in the same process, if there is a adenine, they attach thymine. If there is a guanine, they attach cytosine and so on. But later what happened? Can you see there are some gaps in between? These gaps are filled with the last enzyme of this process. Do anyone know the enzyme name? The enzyme which is sticking or which is attaching each of this Okazaki fragment is known as ligase. And what work it is doing? It is gluing all the Okazaki fragment together. Therefore, it is known as a gluing enzyme. And the name of this gluing enzyme is ligase. The name of this gluing enzyme is ligase. Now this ligase bind all the broken part or the Okazaki fragment together using phosphodiester bond, and this is how we get another new strand. But the problem is it is not leading like the first time. It is lagging, right? It is creating in fragments and then joining together. That's why it is known as lagging strand. Now here, do you remember in the beginning I asked you a question? Why someone compliment us that the eyes of somebody look like mother or father or maybe of like a grandfather, or why are some of the characteristic match with our ancestors or maybe our closest relative or maybe our father or mother? The reason is this. Can you see? Whenever a new DNA is formed, one parental strand and one new strand is formed together, right? So every time this replication process is happening, one parental strand is conserved and a new strand attached. That's why it is known as semi-conservative in nature. Because of one parental strand and one new strand, we all have some kind of characteristic which match to our parents. And that's the reason, or that's the end of the story, why we have some or the other thing which match to our parents. So next time anyone compliment you that why your eyes or you have such a pretty eyes like your mother, you can say because of DNA replication. Okay. So to make you understand in a better way, I have one animation for you, which I will show now.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay. Okay, guys, I hope so you understood this topic very nicely. If you have any question, you can feel free to ask me. Till that time, take care. Bye-bye.
Here is a cell